Morning, this is Ms. Kazishki, and I'm going to talk about adaptive immunity today. I'm going to start using this piece of paper, so if you'd like to follow along by writing, you can on your own paper. And then I'm going to move to this diagram, which I linked in the calendar if you have access to a printer and would like a copy for yourself. Adaptive immunity is our specific immunity, so it's not something we're born with. It's something we build up as we are exposed to pathogens in our environment. And adaptive immunity, first of all, is run by lymphocytes. Lymphocyte means a type of white blood cell, so it's a type of leukocyte. And the two types of lymphocytes are T cells and B cells. You can see that there's two types of T cells and then B cells. And the reason why they have different names is because of where they mature, discussed by Mrs. Komarnicki last week. But they also have uniquely shaped receptors. So these are surface molecules on the outside of cells that are stuck to the membrane. And this would be like one receptor shape, whereas different cells are going to have different receptor shapes. So this is shaped like a U. Another cell, lymphocyte, could have a receptor that's shaped kind of like a V. So a pointed molecular shape is different from this curved or round molecular shape, and that's what makes those receptors unique and able to recognize and respond to different pathogens. Point I want to make here is helper T is the star of the show, okay? Helper T is the major orchestrator of the adaptive immune response. The helper T, once activated, will activate B cells and will activate cytotoxic T. But this guy is at the center of the stage, the main character in the show, and is going to be, like I said earlier, the orchestrator of adaptive immunity. Okay, next, main idea number 2. Lymphocytes can't respond to pathogens directly. So these cells up here can't identify a pathogen and attack it without something called antigen presentation. And antigen presentation you learned about last week is simply the idea of a macrophage gobbles up a pathogen by phagocytosis and presents a piece of that pathogen on its surface. So a macrophage is like that big eater, gobbles up the pathogen. You'll see a picture of that in a minute on the other slide. The macrophages present antigens to lymphocytes. These are lymphocytes. Now, because the macrophage is a phagocyte, sometimes we call the macrophage a phagocyte. I should have said, because the macrophage performs phagocytosis, we call the macrophage a phagocyte. It's a type of white blood cell, so it's also a leukocyte, and it's an antigen presenting cell. And so these terms, they get annoying, right? Because they overlap each other, and there's categories of terms within other categories of terms, but hopefully this clarifies things for you. Third main idea is this is going to be an attack on two fronts, meaning there's two main strategies that the adaptive immune response utilizes. And it's because the pathogens exist in two locations. So the humoral response we're going to talk about tomorrow, not today, but this is run by B cells. And this is an attack on free pathogens, as in pathogens floating around in your blood, your saliva, in your intestines, pathogens that are in bodily fluids. And then the cell-mediated response, which is what I'm going to talk about today, is run by cytotoxic T cells. And this is going to be pathogens and I'm going to abbreviate here, inside your body cells. So if we just take the coronavirus, the virus is going to be found free or unattached to cells in the human body in a person who's infected. And it's also going to be found inside body cells, particularly the cells of the lungs. And so because these pathogens now exist in 2 locations, 2 different types of attacks are going to have to be launched. Both of these require an active helper T cell. Okay, so now we're going to move to this paper, and you're going to see that these images, they're a little cartoony, but what you can see here are the pathogens. So this is a pathogen. Here's pathogens floating around in bodily fluids. And here's pathogens inside body cells. So this could be a lung cell, this could be a liver cell, this could be a skin cell. It could be any type of cell in a human that has been attacked and now harbors the pathogen inside it. And what the immune system needs to do is obviously kill these, get rid of them, but the immune system also has to kill infected body cells. So this is a cell that normally we want to keep because it's a human cell performing a certain function. But in this case, this guy's got to go, okay? He's sad, wants to die. So he's going to go. Now, antigen presentation is the first step in both of these. And antigen presentation happens when a pathogen enters the body and gets swallowed up by a macrophage, okay? So this is otherwise known as phagocytosis. That's the process. And what happens when this guy is gobbled up by the macrophage is he gets chewed up into pieces. And some of those pieces get presented, put a few more circular ones in there, get presented on a surface molecule. So this is the antigen, which corresponds to the surface identity molecule of the pathogen. And this macrophage here is presenting the antigen. Who's he presenting the antigen to? A helper T cell with a receptor that matches the shape of the antigen. So the fact that this is curved signifies that it matches the shape of the unique antigen on the surface of the pathogen we are responding to, helper T. And here I'm just going to add that in written form, this T cell receptor matches antigen shape. It's really important. So if you have the coronavirus, hoping you don't, right? But if you do, there's going to be those spikes that you see in the diagrams, and those spikes are unique to that virus. So the antigen on the coronavirus is different from the antigen on any other virus. And so this specificity is important to the adaptive immune response. Excuse me. This up here, also just side note, this line represents any sort of barrier. So we talked about that last week. Like the skin would be a barrier, your saliva would be a barrier, enzymes or hostile chemicals anywhere in your body would be a barrier. So here we have the pathogen achieving entry over that barrier, and we know that the barrier is part of the innate immune response, whereas this is adaptive. Okay, so what's our goal? Our goal is for the cell-mediated response, we got to kill this guy. Okay, why? Because he can't function, this cell can't function as it normally would. It's infected. It literally has the pathogen inside it. And in the case of viruses, viruses use host cells to make more copies of themselves. So what we want to do is get rid of the body cell that the virus is using as its little factory. And the way we do that is, like I said before, the helper T, now active, I'm switching colors here, is going to release a new molecule called a cytokine, or plural cytokines. And so those cytokines, again, they're only produced and released after antigen presentation. So after activity is generated by the recognition of the antigen presented to the helper T, these cytokines are going to activate another molecule, another cell type, and that cell type is the cytotoxic T. Cytotoxic literally means toxic to cells. Okay, so it is a cell. It's an immune system cell. It's a lymphocyte. Specifically, it's a T cell, but it's going to be toxic to other cells. So remember our goal. Our goal is to kill this guy, infected body cell. The cytotoxic T is going to do that. So the cytokines are going to activate the cytotoxic T. The cytotoxic T happens to have the exact same receptor shape. So this receptor matches the shape of this receptor. So these T cells have unique shapes. When compared to each other, they're the same, but when compared to other varieties of T cells, they're going to be uniquely shaped for this pathogen. And the way that the cytotoxic T knows that a cell is infected, like the way that this guy knows to kill this guy, is antigen presentation again. So here we have an antigen on the surface of a pathogen, infected body cells, whether they be lung cells or any other cell type, skin cell, digestive cell. Infected body cells break up pathogens and they present them much like the macrophage did. But here, when the macrophage is presenting the antigen to the helper T, the macrophage is saying, I'm on surveillance. I'm letting you guys know that I caught a bad guy. This is what the bad guy looks like. Go do something about it. Over here, this is antigen presentation as well. This cell is presenting the antigen to the cytotoxic T, saying, hey, I'm over here, I'm the one, or we are the ones, these cells in this part of the body are the ones that need to go. Like, I'm hurting, you need to kill me, here's the pathogen that's hurting me, and because you have the same matching receptor shape, you can identify me. So that is it for today. We have reviewed adaptive immunity. We've looked at the two types of attack. Attack over here, that's going to be for tomorrow. This was how do we get body cells that are infected out, and we kill them with cytotoxic T cells that require activation from helper T cells that require activation by antigen presentation. You can imagine if you're losing a lot of infected body cells, like if this guy's dying, that's not going to feel so good. So symptoms like cough and difficulty breathing, again, I'm talking about COVID, but symptoms of being sick are oftentimes a result of the immune system working rather than the virus itself causing those symptoms. I know it's hard to imagine because we think of the virus as the enemy, and it is. But when the immune system responds to pathogens, it gets extremely active and you start losing body cells. And these chemicals, while they do activate the cells that protect you, they can increase inflammation and difficulty with normal cell function. So that's it. Thank you for listening. If you have any questions, feel free to email me. Tomorrow we will hit this side. So this is cell-mediated response, okay? Over here, humoral response. All right, thank you. Bye-bye.